Welcome to the deep dive. If you've been following our discussions, you know the premise here. We take a stack of critical source material.
And we extract the absolute core of what really matters.
Right. We strip away all the noise. And today, our mission is to build a rigorous understanding of a foundational topic for medical aspirants, which is Chapter 2, Sexual Reproduction in Flowering Plants.
Yes. Angiosperms.
Yeah, angiosperms. So let's just jump right in because I had this massive breakthrough just reading the introduction to our source text today. It does something highly unusual for a biology manual.
Oh, so.
It opens with what is basically an evolutionary reality check for you, the reader. It points out how we as humans, we swoon over the scents, the perfumes, the rich colors of flowers. We assume nature produced these masterpieces just for our aesthetic pleasure.
Right, the classic human ego.
Exactly. But then the text clearly states, and this is a quote, that flowers do not exist for us or to be used for our own selfishness.
That is a harsh truth.
It really is. Every time you stop to admire a perfectly manicured botanical garden, if you're viewing it through that human-centric lens, you are entirely misunderstanding the biology happening right in front of you.
It is a brilliant way to open a highly technical text because, it forces an immediate paradigm shift. If you are preparing for a rigorous, high-stakes exam like Neet, you just cannot afford to look at botany through an anthropocentric lens.
You have to be totally objective.
Yes, you have to evaluate these structures strictly on their evolutionary return on investment. Today, our goal is to build an unshakable mental map of how angiosperms actually work.
Not just memorizing a table of contents.
Exactly. We are going to dissect why these mechanisms exist, the specific problems they solve in nature, and exactly how examiners use the structure of this chapter to test your biological intuition.
That evolutionary ROI, that's the perfect framework. The text immediately defines the core concept here. Every single visual and olfactory beauty, the vivid petals, the nectar is strictly an aid to sexual reproduction.
Nothing more, nothing less.
Right. And since we already know basic floral morphology, sepals, petals, stamens, carpels, let's look at how the text links those structures directly to their function. Building a massive corolla or synthesizing complex, volatile organic compounds for scent, that is a massive metabolic drain on the plant.
It's extremely expensive.
Imagine walking into a high-stakes, multi-billion-dollar corporate negotiation, seeing a lavish buffet, and assuming the entire company was built just to give you a free lunch.
That is exactly how humans treat flowers.
Right. The plant expends this metabolic energy. It blows half its annual marketing budget because the return, which is successful sexual reproduction, is the absolute priority for the survival of the lineage.
And that leads directly to a universal truth stated in the intro that you need to anchor in your mind right now. The text explicitly says all flowering plants show sexual reproduction.
All of them.
Yes. When a syllabus or a core scientific text uses an absolute term like all, you need to pay attention. In biology, we are so conditioned to look for exceptions, and we will actually cover some highly testable exceptions later in this deep dive.
Oh, yeah, the rule breakers.
Right. But right here, the text establishes a non-negotiable baseline.
Right.
If it is an angiosperm, sexual reproduction is its defining characteristic. Examiners love testing absolutes because students often second-guess themselves. They sit there wondering if there is some obscure grass species out there that doesn't fit the rule.
They overthink it.
Exactly. But for the purpose of the foundational definition, the text says all.
So it provides this totally solid baseline before we get into the complex physiology. Looking at the structural flow of the chapter, sections 2.1 through 2.5, it's clear this isn't just a random assortment of botanical facts thrown together.
No, it is a highly structured narrative.
It maps out a logical, chronological timeline of a plant's reproductive life. We start with the organ itself in 2.1. We move to the pre-fertilization prep in 2.2, hit the signature main event of double fertilization in 2.3.
The big show?
The big show, yeah. Then we deal with the post-fertilization aftermath in 2.4, and finally, look at those specialized rule breakers in 2.5.
That chronological flow is your ultimate guide as a learner. It's your mental timeline. You build the stage, you prepare the actors, you execute the climax, you secure the legacy, and then, only then, you study the avant-garde alternative methods.
That makes so much sense.
And the author also drops a critical triad in the introduction that should govern how you study every single one of these sections: morphology, structure, and process.
I noticed that. The final sentence of the intro explicitly mandates that we understand the morphology, structure, and the processes. It's basically telling you, do not try to memorize the physiological process without first understanding the physical anatomy.
You cannot build the roof before the foundation.
Right. Morphology is observing the external form, the arrangement of the whorls. Structure, or anatomy, is the internal histological organization, like the layers of the anther wall or the cellular layout of the embryo sac. And the process is the physiology, microsporogenesis, the actual growth of the pollen tube.
And this is precisely where so many students hit a wall. They try to rote learn the steps of syngamy without a solid grasp of the embryo sac's internal architecture.
Which just leads to confusion.
It becomes a fragile memorization of facts rather than a robust, intuitive understanding. The text is warning you early on. Master the form before you attempt to master the function.
So let's apply that framework to section 2.1, which focuses on the flower as a fascinating organ. The text highlights the sheer diversity of inflorescences, flowers, and floral parts.
The massive range.
It is. When we consider the variation from a wind-pollinated grass flower that is visually, totally unremarkable, to the highly complex, specialized orchid, the underlying theme is adaptation. And the source explicitly states the biological purpose behind this amazing range of adaptations. It is to ensure formation of the end products of sexual reproduction.
Which are the fruits and seeds. This cannot be overstated for your exam prep. Every single anatomical variation, every hormonal shift that triggers the shoot apical meristem to transition into a floral meristem is biologically calculated to secure those end products.
It all comes back to the seeds.
Always. A flower that fails to produce a viable seed is an evolutionary dead end. When you face a conceptual question asking why a specific floral adaptation provides an advantage, say, the synchronization of pollen release and stigma receptivity, the core of the correct answer will almost always tie back to maximizing the probability of forming high-quality fruits and seeds.
That sets up the stakes perfectly. So the decision is made at the hormonal and structural level, the floral primordium develops, and we transition into section 2.2. Pre-fertilization, structures, and events. It's a crucial distinction the syllabus makes right there in the heading.
Yes, separating the physical from the functional.
Right. We aren't just looking at the overarching phase. We are explicitly separating the physical architecture, the structures, from the preparatory actions, the events.
And let me tell you, this is a classic trap in multiple choice questions.
Yeah.
You will be asked to identify a pre-fertilization structure, and the options will mix anatomical terms with physiological processes.
Oh, that's sneaky.
Very. If you choose pollination when asked for a structure, you lose the marks, even though pollination occurs during the pre-fertilization phase. The examiners are testing your precision. So let's break down the male reproductive whorl first.
The endothecium.
Correct. The endothecium consists of stamens, and each stamen has the long, slender stalk called the filament, and the terminal, typically bilobed structure called the anther.
And the syllabus gets very specific about the morphology of that typical angiosperm anther. It describes it as bilobed, with each lobe having tooth-decay, making it dithecous. So if you do a cross-section, it reveals a four-sided or tetragonal structure consisting of 4 microsporangia located at the corners.
2 in each lobe.
Exactly. This is where we bridge from morphology into internal structure.
So let's go deeper into that internal structure, specifically the microsporangium wall, because this is extremely high yield for exams. A typical microsporangium appears near circular in outline and is surrounded by 4 distinct wall layers.
I know the layers.
Absolutely. From the outside in, you have the epidermis, the endothecium, the middle layers, and finally the innermost layer, the tapetum. The outer 3 layers are primarily mechanical. They perform the function of protection and help in the dehiscence of the anther to release the pollen. But the tapetum...
The tapetum is the biological VIP here.
It really is.
It's fascinating because its physiological role is so active. While those outer layers are just structural, the tapetum is actually responsible for nourishing the developing pollen grains. The text notes that cells of the tapetum possess dense cytoplasm and generally have more than one nucleus.
And that multinucleate condition is a frequent target for examiners.
Yeah.
They will ask you why. Why is it multinucleate?
Right, because of endomitosis.
Exactly. Endomitosis, or free nuclear division without cytokinesis. The tapetum needs a massive amount of metabolic machinery to synthesize the nutrients, hormones, and specialized proteins required for pollen development. It's essentially the kitchen for the developing pollen.
And it also produces eubish bodies, right?
Yes, which are crucial for the formation of the outer layer of the pollen grain. But let's not forget what is sitting right in the center of this protective, nourishing structure, the sporogenous tissue.
Right in the middle. When the anther is young, this compact group of homogeneous cells occupies the center of each microsporangium, and as the anther develops, these cells undergo microsporogenesis. This is our first major event under the pre-fertilization heading.
Structure to event.
Yep. The cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. And since each cell of the sporogenous tissue is capable of giving rise to a tetrad, each one is a potential pollen mother cell, or PMC.
The transition from PMC to microspore tetrad is meiosis. That means we are moving from a diploid state 2N to a haploid state N.
That ploidy shift is fundamental.
It is arguably the most important thing to track. Students must track the ploidy level of every structure we discuss. Epidermis, 2N and tapetum. Polyploid or multinucleate, but originating from 2N tissue. Pollen mother cell, 2N microspore NN. The process of forming these haploid microspores from a diploid PMC through meiosis is the strict definition of microsporogenesis.
So, as the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Which brings us to the structure of the pollen grain itself, the male gameporphyte. And this isn't just a simple sack of DNA. It has a highly stratified, incredibly resilient architecture.
Nature's armored transport.
Exactly. The pollen grain has a prominent two-layered wall. The hard outer layer is the exine, and this is where we encounter one of the most remarkable substances in the entire plant kingdom, sporopollenin.
Sporopollenin is an absolute marvel of evolutionary engineering, and I can tell you it is arguably one of the most heavily tested molecules in botany. It is one of the most resistant organic materials known to science. It can withstand high temperatures, strong acids, and strong alkalis.
Basically indestructible.
Essentially, no enzyme that degrades sporopollen has been discovered so far.
And that resistance is exactly why pollen grains are so well preserved as fossils. It's nature's ultimate biological armor. But that raises a question. If the pollen grain is completely encased in an impenetrable shield, how does the genetic material ever get out to actually perform fertilization?
That brings us to a beautiful structural nuance. The exine is not a continuous, unbroken sphere. It has prominent apertures called germ pores, where sporopollenin is completely absent.
A built-in exit hatch.
Precisely. It is a brilliant evolutionary compromise. You have maximum armor for the perilous journey of pollination, but a designated unarmored exit hatch to allow the pollen tube to emerge when it lands on a compatible stigma.
And beneath that exine is the intine.
Right, which is a thin, continuous layer made up of cellulose and pectin. It's the intine that will eventually elongate to form that pollen tube.
Okay, so inside the mature pollen grain, we see further cellular division. The microspore nucleus divides mitotically to form two distinct cells, the vegetative cell and the generative cell. The vegetative cell is massive. It has abundant food reserve and a large, irregularly shaped nucleus. It essentially acts as the life support system and the logistical engine for the eventual pollen tube.
And the generative cell.
The generative cell is small, spindle-shaped, and it actually just floats in the cytoplasm of the vegetative cell.
And here is a critical data point that frequently appears on exams. In over 60% of angiosperms, pollen grains are shed at this exact two-celled stage.
But what about the other 40%?
In those species, the generative cell divides mitotically before the pollen is shed, giving rise to the two male gametes. So the pollen is shed at a three-celled stage. You must remember the 60-40 split and the difference in cellular composition at the time of shedding.
So just to track the ploidy and the cellular count again. A pollen grain shed at the two-celled stage contains one vegetative cell, which is N, and one generative cell, also N. The three-celled stage contains one vegetative cell, N, and two male gametes, both N. The generative cell is basically the payload, and the vegetative cell is the delivery vehicle.
A perfect way to visualize it.
Now, the text also notes the real-world implications of pollen, because it's not just an academic concept. Many species, like Parthenium or carrot grass, which came into India as a contaminant with imported wheat, they cause severe pollen allergies and bronchial afflictions like asthma and bronchitis.
It highlights the sheer volume and environmental presence of pollen. It's everywhere. But from a botanical standpoint, once pollen is shed, it's a race against the clock. Pollen viability is highly variable.
It depends on the species, right?
Heavily. In some cereals like rice and wheat, pollen grains lose viability within 30 minutes of release. Half an hour, and they're done. In members of Rosaceae, Leguminosae, and Solanaceae, they maintain viability for months.
Which ties back to the storage of pollen. Just as we have sperm banks for humans, we can store pollen grains of a large number of species for years in liquid nitrogen at minus 196 degrees Celsius.
Cryopreservation.
Yeah. These pollen banks are critical resources in crop breeding programs.
So that covers the male pre-fertilization structures and events extensively. Now we have to shift our focus to the female reproductive world, the gynoecium.
The female reproductive part of the flower. And the terminology here really builds in complexity. We start with the pistil. A gynoecium can be monocarpellary, having a single pistil, or multicarpellary. If there are multiple pistils, they can be fused together, which is syncarpous, or free, which is apocarpous.
And each pistil has three parts.
Right. The stigma, the style, and the ovary.
The stigma serves as the landing platform for pollen grains. The style is the elongated, slender part beneath the stigma. But the basal, bulged part is the ovary, and this is where the critical pre-fertilization events occur. Inside the ovarian cavity, or locule, is the placenta. Arising from the placenta are the microsporangia, commonly called ovules.
Let's look at the structure of a typical anatropous ovule, because this is another heavily tested anatomical diagram. You always see this in the papers. The ovule is attached to the placenta by a stalk called the funicle. The body of the ovule fuses with the funicle in the region called the hilum. So the hilum represents the physical junction between the ovule and the funicle.
Good. And protecting the ovule are one or two protective envelopes called integuments. These integuments encircle the entire ovule except at the tip, where a small opening called the micropyle is organized.
The micropylar end.
Yes. And opposite the micropylar end is the chalaza, representing the basal part of the ovule. This polarity micropylar versus chalazal is essential to master, because the entire development of the female gamophyte is oriented along this exact axis.
So enclosed within the integuments is a mass of cells called the nucellus. The cells of the nucellus have abundant reserve food materials. And just like the sporogenous tissue in the anther, the nucellus is the starting point for gamete formation. This is where we transition from structure to the event of microsporgenesis.
The parallel to the male side is very clear here.
It really is. The process begins with a single cell. The ovule generally differentiates a single megaspore mother cell, or MMC, in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus.
Notice that parallel. We had a pollen mother cell, tucken, undergoing meiosis. Here we have a megaspore mother cell, tucken, undergoing meiosis. The MMC undergoes meiotic division to produce 4 haploid megaspores. However,
That's the catch.
right, unlike the male side, where all 4 microspores typically survive to become pollen grains, the female side is much more resource-intensive and selective. In the majority of angiosperms, one of the megaspores is functional, while the other 3 degenerate.
Only one survives.
Only the functional megaspore develops into the female gamophyte, which is the embryo sac.
And this specific method of embryo sac formation from a single megaspore is termed monosporic development. The ploidy tract is crucial here too. The nucellus is tucken, the MMC is tucken, the functional megaspore is N, and the resulting female gamophyte is N.
The events that follow the selection of the functional megaspore are fascinating because it involves sequential nuclear divisions without immediate cellularization.
Free nuclear division.
Yes, the nucleus of the functional megaspore divides mitotically to form 2 nuclei, which move to opposite poles, forming the 2-nucleate embryo sac. 2 more sequential mitotic nuclear divisions result in the 4-nucleate and later the 8-nucleate stages of the embryo sac.
And like you said, these mitotic divisions are strictly free nuclear, meaning nuclear division is not immediately followed by cell wall formation. It's only after the 8-nucleate stage that cell walls are laid down, leading to the organization of the typical female gametophyte.
The structural organization of this mature embryo sac is a masterpiece of biological architecture, and it is guaranteed to be tested. Out of the 8 nuclei, 6 are surrounded by cell walls and organized into distinct cells. Let's map them out along that axis we mentioned earlier.
Okay, chalazal versus micropolar.
At the chalazal end, we have 3 cells called the antipetals. At the micropolar end, 3 cells are grouped together to form the egg apparatus.
And the egg apparatus is the primary target for fertilization. It consists of 2 synergids and 1 central egg cell. The synergids are not just space fillers. They have a highly specialized cellular thickening at the micropylar tip called the filiform apparatus.
A very important structure.
Yeah, this structure plays the vital role of guiding the pollen tube exactly where it needs to go. It's like a botanical beacon.
And so that accounts for 6 cells.
Right.
But what about the remaining 2 nuclei?
They are called polar nuclei, and they don't get their own individual cell membranes. Instead, they are situated together in the massive central cell right below the egg apparatus. This means a typical angiosperm embryo sac, at maturity, though it is 8-nucleate, is actually only 7-celled.
8-nucleate, 7-celled. That phrase should be permanently etched into your memory. It is a defining structural characteristic of the female gametophyte in typical angiosperms. You have 3 antipetals, 2 synergids, 1 egg cell, and 1 large central cell containing 2 polar nuclei.
So we've now built the pollen grain and we've built the embryo sac. The actors are fully prepped, but they are physically separated. In angiosperms, both male and female gametes are non-motile. They have to be brought together for fertilization to occur, which brings us to the pivotal event bridging pre-fertilization and the main event, pollination.
The transfer.
Right. The text defines pollination simply as the transfer of pollen grains from the anther to the stigma of a pistil. But the mechanisms are incredibly diverse.
Depending on the source of the pollen, pollination can be divided into 3 distinct types: autogamy, geitonogamy, and xenogamy.
Let's start with autogamy.
Autogamy is pollination within the same flower. For autogamy to occur, you need synchrony in pollen release and stigma receptivity, and the anther and stigma must lie close to each other. Some plants, like viola, oxalis, and commelina, take this a step further by producing 2 types of flowers: chasmogamous flowers, which have exposed anthers and stigmas like typical flowers, and cleistogamous flowers, which do not open at all.
Cleistogamous flowers are a brilliant evolutionary failsafe. In such flowers, the anthers and stigma lie very close to each other inside the closed bud. When anthers dehisce, the pollen falls directly on the stigma. Cleistogamous flowers are invariably autogamous because there is 0 chance of cross-pollen landing on the stigma.
It's physically impossible.
Yeah, the text points out that they produce assured seed set even in the absence of pollinators. It's a guaranteed return on investment, though it comes at the cost of genetic diversity.
And that cost is significant in evolutionary terms, which is why most plants prefer cross-pollination. So the second type is geitonogamy, the transfer of pollen from the anther to the stigma of another flower on the same plant.
So it's ecologically cross-pollination, but genetically self-pollination.
Exactly. Ecologically, it requires a pollinating agent, so it functions like cross-pollination. But genetically, it is similar to autogamy because the pollen comes from the same plant. Finally, we have xenogamy, the transfer of pollen from the anther to the stigma of a different plant. This is the only type of pollination that brings genetically different types of pollen grains to the stigma.
To achieve geitonogamy and xenogamy, plants use agents of pollination grouped broadly into abiotic wind and water and biotic animals. The adaptations for wind pollination, or anemophily, are distinct. Wind-pollinated flowers need to produce enormous amounts of pollen because the process is entirely random and highly wasteful.
It's a numbers game.
Totally. The pollen grains are light and non-sticky, so they can be transported by wind currents. They often possess well-exposed stamens, so the pollen is easily dispersed, and large, feathery stigmas to easily trap airborne pollen.
A classic example of a wind-pollinated structure is the tassel of a corncob. Those are the stigmas and styles waving in the wind. Wind pollination is quite common in grasses. They usually have a single ovule in each ovary and numerous flowers packed into an inflorescence. They don't waste energy on colorful petals or nectar because the wind doesn't care what the flower looks or smells like.
The wind has no eyes. Now, water pollination, or hydrophily, is quite rare. It's limited to about 30 genera, mostly monocotyledons like Vallisneria and Hygrilla in freshwater, and marine seagrasses like Zostera.
The mechanism in Vallisneria is particularly elegant.
It is. In Vallisneria, the female flower reaches the surface of the water by a long stalk, and the male flowers or pollen grains are released onto the surface of the water. They are carried passively by water currents. In seagrasses, the female flowers remain submerged, and the pollen grains are long, ribbon-like, and carried passively inside the water.
And to protect against getting wet, pollen grains in most water-pollinated species are covered by a mucilaginous covering.
But the vast majority of flowering plants rely on biotic agents, animals, bees, butterflies, flies, beetles, wasps, ants, moths, and even birds and bats. Insect-pollinated flowers, or entomophilous flowers, are the ones that deploy that massive corporate marketing budget we discussed earlier.
They pull out all the stops.
They are large, colorful, fragrant, and rich in nectar. If the flowers are small, they are clustered into an inflorescence to make them conspicuous. The rewards are the critical part of the transaction. Nectar and pollen grains are the usual floral rewards for the pollinators.
But sometimes the reward is providing a safe place to lay eggs.
Right. The tallest flower of a Morphophilus provides this, as does the fascinating relationship between a species of moth and the plant yucca. The moth deposits its eggs in the locule of the ovary, and the flower gets pollinated in return. The larvae come out of the eggs as the seeds start developing. They cannot complete their life cycles without each other.
It's a perfect symbiosis. Now, because continued self-pollination results in inbreeding depression, flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination. These are known as outbreeding devices.
How do they do it?
Well, in some species, pollen release and stigma receptivity are simply not synchronized. Either the pollen is released before the stigma is receptive, or the stigma becomes receptive long before pollen release. In other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower.
Another fascinating outbreeding device is self-incompatibility. This is a genetic mechanism that prevents self-pollen, whether from the same flower or other flowers of the same plant, from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil.
It's essentially a biochemical lock and key system that rejects genetic matches.
Another approach is the production of unisexual flowers. If both male and female flowers are present on the same plant, like in castor and maize, which is monoecious, it prevents autogamy but not diatonogamy. But if male and female flowers are present on different plants, like papaya, so dioecious, it prevents both autogamy and diatonogamy.
And all of these mechanisms lead to the ultimate pre-fertilization test, pollen-pistil interaction. Pollination doesn't guarantee the transfer of the right type of pollen. The pistil has the ability to recognize the pollen, whether it is compatible or incompatible.
It's a chemical dialogue.
Exactly. This recognition is the result of a continuous chemical dialogue mediated by chemical components of the pollen interacting with those of the pistil. If it is compatible, the pistil accepts the pollen and promotes post-pollination events.
So the compatible pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. The contents of the pollen grain, the vegetative nucleus, and the two male gametes, if it was shed at the three-celled stage, or the generative cell, which will divide in the tube if shed at the two-celled stage, move into the pollen tube. The tube grows through the tissues of the stigma and style and reaches the ovary.
And here we reconnect with the anatomy we mapped out earlier. After reaching the ovary, the pollen tube enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus.
The filiform apparatus actively guides the entry of the pollen tube. All of these events, from pollen deposition on the stigma until pollen tube enters the ovule, are collectively referred to as pollen-pistil interaction. It is a dynamic process involving pollen recognition followed by promotion or inhibition.
And this precise guidance by the filiform apparatus sets the stage perfectly for section 2.3. Double fertilization.
The main event.
When we look at the timeline of the chapter, pre-fertilization covers a massive amount of structural development and events. Post-fertilization covers the entire aftermath of seed and fruit development. But right here in the middle, double fertilization gets its very own standalone section. It is given equal billing with entire phases of life.
Why does it get its own section? Why not just call it fertilization?
That standalone heading is a massive signal to you, the student. When a specific named physiological process is elevated to the level of a major structural section, it means it is the defining high-yield characteristic of the entire clade. Double fertilization is the signature move of angiosperms. It is entirely unique to them.
You won't see this in gymnosperms.
No, you won't. The text separates it because understanding the specific mechanics of this event is fundamental to understanding everything that follows.
So let's break down the mechanics. The pollen tube has entered one of the synergids, and it releases the two male gametes into the cytoplasm of the synergid. We have two male gametes, which immediately explains the double in double fertilization. They don't just act as backups for each other. They take two entirely different paths to accomplish two distinct fertilization events.
Let's follow the first male gamete. It moves towards the egg cell and fuses with its nucleus, completing syngamy. The result of syngamy is the formation of a diploid cell, the zygote 2M8. This is standard sexual reproduction. The zygote will eventually develop into the embryo, the next generation of the plant.
But then we have the second male gamete. It doesn't target the egg. Instead, it moves towards the two polar nuclei located in the massive central cell. It fuses with them to produce a triploid primary endosperm nucleus, or PN. Because this fusion involves three haploid nuclei, one from the male gamete and the two polar nuclei, it is termed triple fusion.
And this is where examiners will test your ability to track ploidy levels under pressure. Syngamy results in a diploid, 2N zygote. Triple fusion results in a triploid, 3N primary endosperm nucleus. Since two types of fusions, syngamy and triple fusion, take place simultaneously in the embryo sac, The phenomenon is termed double fertilization.
So the central cell after triple fusion becomes the primary endosperm cell, the PEC, and develops into the endosperm, while the zygote develops into an embryo.
Exactly. And you have to understand the elegance of this system. It is stunning. In many gymnosperms, the plant develops massive amounts of nutritive tissue before fertilization even occurs.
Just hoping it gets fertilized.
Yes, and if the seed isn't fertilized, all that energy is wasted. But angiosperms use double fertilization to ensure that the massive investment in nutritive tissue, the endosperm, is only initiated after the egg is successfully fertilized.
So the triple fusion acts as a developmental trigger.
Precisely. It perfectly aligns resource expenditure with reproductive success. It is the ultimate expression of metabolic efficiency.
Which perfectly transitions us into section 2.4, post-fertilization, structures and events. Following double fertilization, the events of endosperm and embryo development, maturation of ovules into seeds, and ovary into fruit are collectively termed post-fertilization events. And here is a crucial sequencing fact that the text emphasizes. Endosperm development precedes embryo development.
The text explicitly asks why, and the answer is rooted in that same metabolic logic we just discussed. The primary endosperm cell divides repeatedly to form the triploid endosperm tissue. The cells of this tissue are filled with reserve food materials and are used for the nutrition of the developing embryo.
So the plant guarantees that the food supply is established before the embryo begins its major growth phases. It's an evolutionary insurance policy.
A very smart one. The development of the endosperm itself is fascinating. In the most common type, the PEN undergoes successive nuclear divisions to give rise to free nuclei. This is called free nuclear endosperm. Subsequently, cell wall formation occurs, and the endosperm becomes cellular.
Is there a good example of this?
Yes, the classic, highly testable example of this is the coconut. The coconut water from a tender coconut is nothing but free nuclear endosperm made up of thousands of nuclei, while the surrounding white kernel is the cellular endosperm.
That real-world connection is perfect. The water is nuclear, the meat is cellular. While the endosperm is providing the nutrition, the embryo is developing at the micropylar end of the embryo sac, where the zygote is situated. Most zygotes divide only after a certain amount of endosperm is formed.
Embryogeny.
Right. The early stages of embryo development, embryogeny, are similar in both dicotyledons and monocotyledons. The zygote gives rise to the proembryo and subsequently to the globular, heart-shaped, and mature embryo.
Let's dissect the mature dicotyledonous embryo first. It consists of an embryonal axis and two cotyledons. The portion of the embryonal axis above the level of cotyledons is the epicotyle, which terminates with the plumule or stem tip.
And the lower part.
The cylindrical portion below the level of cotyledons is the hypocotyle, which terminates at its lower end in the radicle or root tip. The root tip is covered by a root cap.
In contrast, embryos of monocotyledons possess only one cotyledon. In the grass family, this single cotyledon is called the scutellum, and it is situated towards one side, or lateral, of the embryonal axis. At its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called the coliorhiza.
And the upper part.
The portion of the embryonal axis above the level of attachment of the scutellum is the epicotyle. The epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. The structural differences, two cotyledons versus a scutellum, the presence of coliorhiza and coleoptile are classic comparative testing points.
As the embryo develops and matures, the overarching structure, the ovule, is also transforming into a seed, and the ovary is transforming into a fruit. This is the fulfillment of the end products we discussed in the very beginning. A mature seed consists of seed coats, cotyledons, and an embryo axis.
The text makes a crucial distinction regarding how seeds manage their endosperm. In non-albuminous or exalbuminous seeds, the endosperm is completely consumed during embryo development before seed maturation. Good examples are pea and groundnut.
And the other type.
In albuminous seeds, a part of the endosperm is retained because it wasn't completely used up during embryo development. Examples are wheat, maize, barley, castor, and sunflower.
Additionally, in some seeds, like black pepper and beet, remnants of the nucellus are also persistent. This residual persistent nucellus is called the perisperm.
That's a huge trap.
It is. Do not confuse endosperm, which is 3N and formed from double fertilization, with perisperm, which is 2N and a remnant of the nucellus. Examiners will absolutely test that distinction. As the seed matures, the integuments of the ovule harden into tough, protective seed coats. The micropyle remains as a small pore in the seed coat to facilitate the entry of oxygen and water during germination.
As the seed matures, its water content is drastically reduced, and the seed becomes relatively dry, dropping to 10–15% moisture by mass. The general metabolic activity of the embryo slows down, and it may enter a state of inactivity called dormancy.
This dormancy is a phenomenal adaptation, allowing the seed to survive unfavorable environmental conditions and disperse across time and space. When favorable conditions, adequate moisture, oxygen, and suitable temperature return, they germinate.
While the ovules mature into seeds, the ovary develops into the fruit. The transformation of ovules into seeds and ovary into fruit proceeds simultaneously. The wall of the ovary develops into the wall of the fruit called the pericarp. Fruits can be fleshy, like guava, orange, and mango, or they may be dry, like groundnut and mustard.
And just as we look closely at how parts develop, we have to look at exceptions in fruit formation. In most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in a few species like apple, strawberry, and cashew, the thalamus also contributes to fruit formation.
These are called false fruits. Most fruits, however, develop only from the ovary and are called true fruits.
And then we have the ultimate exception in fruit development, fruits that develop without fertilization. These are called parthenocarpic fruits. The banana is a classic example. Parthenocarpy can also be induced through the application of growth hormones, resulting in seedless fruits. This is highly relevant for agriculture and commerce.
Which perfectly sets up Section 2.5, apomixis and polyembryony. The timeline from 2.1 to 2.4 gave us this beautiful logical progression building up to a seed and a fruit through sexual reproduction. And then at the very end of the chapter, the syllabus introduces these specialized terms.
Their placement at the absolute end signifies that they defy the neat, tidy rules established in the previous sections.
They break the rules.
Yes. This is where we separate the average students from the top-tier candidates. Examiners adore exceptions because they test the absolute limits of your understanding. You cannot comprehend the disruption of a process if you do not master the normal process first. Apomixis is a form of asexual reproduction that mimics sexual reproduction.
Mimicking sexual reproduction is such a specific phrase. Although seeds in general are the products of fertilization, a few flowering plants, such as some species of Asteraceae and grasses, have evolved a special mechanism to produce seeds without fertilization. This is apomixis, specifically agamospermy. It results in the formation of clonal seeds.
How does it happen? There are several ways. In some species, the diploid egg cell is formed without reduction. Division meaning meiosis failed to occur or was bypassed, and it develops into the embryo without fertilization. It was already techonent, so it acts like a zygote. More often, as in many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac start dividing, protrude into the embryo sac, and develop into embryos.
Wait, if multiple nucellar cells start developing into embryos within a single ovule, that means a single seed will contain multiple embryos.
Exactly, and that is the strict definition of polyembryony, the occurrence of more than one embryo in a seed. If you squeeze a seed of an orange, you might find many embryos of different sizes and shapes. Because these embryos are derived from the maternal nucellar tissue, which is tocans, they are genetically identical to the parent plant. They are clones.
The text notes a massive real-world application for apomixis. The cultivation of hybrids has tremendously increased agricultural productivity, but the problem is that hybrid seeds have to be produced every year. If the seeds collected from hybrids are sown, the plants in the progeny will segregate and lose the hybrid characters. Production of hybrid seeds is costly, making them expensive for farmers.
But if these hybrids are made into apomix, there is no segregation of characters in the hybrid progeny. The farmer can keep using the seeds raised from his own hybrid crop year after year without having to buy new seeds. The active research into the genetics of apomixis to transfer apomictic genes into hybrid varieties is one of the most important frontiers in modern agricultural science.
It's incredible. The syllabus ends on this point because it bridges pure botanical theory with critical human survival. It's an incredible narrative arc. We started with the basic morphology of a flower, broke down the cellular architecture of the gametes, mapped out the precise physiological events of pollination and double fertilization, saw the development of the final products, and ended on the genetic frontiers of agriculture. Let's synthesize this massive amount of information. Before we wrap up this deep dive, we need to anchor the key takeaways.
Indeed. We have explored the deep physiology and the strategic structure of this chapter. Here is what you must remember to build a resilient foundation for your exams. These are 5 critical memory anchors based on our deep dive. Number 1, the absolute baseline. All flowering plants, the angiosperms, reproduce sexually. When a foundational text uses absolute terms, lock them in. Sexual reproduction is the defining non-negotiable characteristic of the clade. Number 2, the evolutionary ROI. Flowers are highly engineered biological machines designed with one singular goal: to ensure the formation of fruits and seeds. Every structural variation, from a vibrant corolla to a cleistogamous bud, is an adaptation to secure those end products. Number 3, the structural timeline. You must learn the timeline in order and distinguish between anatomy and physiology. Pre-fertilization is about building the structures: microspores, embryo sac, and executing the transport event, which is pollination. Morphology must precede process in your study plan. Number 4, double fertilization is the high-yield signature. It is unique to angiosperms. You must track the ploidy. Singamy produces a 2N zygote, while triple fusion produces a 3N primary endosperm nucleus. The 3N endosperm develops first to guarantee nutrition for the 2N embryo.
And the last one.
Number 5, the rule breakers test your depth. Apomixis, which is seeds without fertilization, and polyembryony, which is multiple embryos per seed, are placed at the end of the syllabus because they are the ultimate test. They mimic or bypass the standard rules, and understanding them is crucial for mastering high-level exam questions and real-world agricultural applications.
Those anchors are perfect. Let me give you the rapid-fire 60-second sticky summary of the whole journey. We are looking at an absolute masterclass in biological survival. The syllabus chapter maps out a chronological drama. First, the plant builds the stage, utilizing immense metabolic energy to construct the anthers and ovules, producing highly protected pollen grains with sporopollenin shields, and the highly organized 7-celled 8-nucleate embryo sac. Then, through the precise mechanics of pollination and the chemical dialogue of the pistil, the actors are brought together. The angiosperm executes its signature climax double fertilization, resulting in a zygote for the future and an endosperm for its immediate food supply. It secures this legacy by packaging the embryo into a resilient seed and often a dispersible fruit. And finally, it bends the rules with exceptions like apomixis, proving that nature will use whatever genetic strategy works to ensure survival. It is morphology, structure, and process integrated flawlessly.
That synthesis covers the breadth and the depth perfectly. You have grasped not just the facts, but the biological logic underlying them. But as we close this deep dive, I want to leave you with one final provocative thought, based on the evolutionary logic we have explored today. We have established that the rich colors and the swoon-worthy scents of typical entomophilous flowers are expensive adaptations specifically designed to attract pollinators.
Right, the massive corporate marketing budget.
So, as you review your notes and observe the natural world, consider the exceptions. What happens to the flowering plants that don't have brightly colored petals or sweet perfumes? Think about the grasses or the massive oak trees. If a flower is completely plain, odorless, and blends in perfectly with the green leaves around it, yet we know from our universal rule that all flowering plants must achieve sexual reproduction to form fruits and seeds. What entirely different invisible strategy must that plain flower be using to achieve that exact same ultimate goal?
That's a great question.
If they aren't spending their energy on a visual and chemical buffet to attract insects, how have they weaponized the abiotic environment, like the wind or water, to do their reproductive labor for them? Keep that invisible force and the anatomical adaptations required to harness it in mind as you explore the incredible diversity of the plant kingdom.